Hi everyone, I'm Sally. Okay, I'm going to give a bit of an overview and I'm kind of taking us, sorry, that was a bit too loud. I'll take us back to some basics. So sorry for those who are already all over phylogenetics. We can talk through things. There are people probably that are far more experts on this than I am, but hopefully it's a good background for some of these analyses to address some of the things that Craig brought up. So just to take us back to thinking about Snip data, and Craig touched on this particularly when thinking about estimating branch lengths. So from our data, is this a laser? There we go. We're getting information about single nucleotide polymorphisms, but often what we're not getting information on is the invariant sites, and this can be particularly important when thinking about branch lengths. So we don't know, for example, if we've got one SNP in 100 base pairs or if we've got one SNP in 1,000 base pairs, and that can have very big impacts, not on the topology necessarily, but on the branch lengths, as Craig highlighted. But there's some real benefits of SNP data. So, looking, as Craig said, with sequencing, we kind of started with mitochondria, amazing, got some phylogenies, but maternally inherited. We realized that having information from multiple nuclear markers can be really powerful because the genome's heterogeneous. So with SNPs, we're getting a lot more representation of independent markers, and because they're SNPs, there's no intralocus recombination. We don't have to phase the haplotypes. So we can get great accuracy for some of the parameters in terms of population size, etc., with more markers. And we get more data for resolving those difficult and/or recent phylogenetic relationships. But we do have to be aware of some of the challenges as well, because sometimes more data is not necessarily that much clearer unless we understand why certain data is different. So we've come in phylogenetics from looking at nuclear sequence data and mitochondrial sequence data to moving towards this sort of SNP revolution, and there's been some neat comparative studies. So here's just an example here of a phylogeny using RAD-seq reduced representation data versus ultra-conserved elements nucleotide sequence data. And broadly, you get concordant topological results. So the data's converging on the same tree. However, what they are finding are differences in these branch lengths. So I guess it depends on what your questions are, as Craig said, in terms of what you're trying to understand. I'm a big believer. I sort of work across the landscape of population genetics to phylogenetics, and I think we shouldn't just be producing a phylogenetic tree. We kind of need to interrogate the evolutionary history, and we can draw upon different analyses. So we can draw upon population genetics analyses to look at migration if we need to. We can look at phylogenies for looking at the relationships. I think we need to apply multiple methods to resolving some of these questions, particularly for complex evolutionary scenarios. Okay, so I'm just going to touch on some of the phylogenetic methods now that are out there. So I don't know, varying levels of expertise around phylogenetics here, but here we have our phylogeny. We're interested in resolving the relationships between lineages. I'll call them lineages rather than species. We don't need to have debates about these things. Or operational taxonomic units, that's a good one. So we're interested in the branch orders, we're interested in the internal nodes, etc. So you've got sort of the distance-based methods, which you estimate a tree with the branch lengths, and they fit a matrix of pairwise distances between the sequences. Then we've got these character-based or tree-searching methods, maximum parsimony. It explains the data with as few evolutionary changes as possible. Largely, people have moved towards using maximum likelihood and Bayesian, where you're using information around a nucleotide substitution model, and given your data, you estimate a likelihood of the tree given the data and the model. And then Bayesian is just an extension of this, where you have a prior probability of parameters and you estimate the most likely tree, and I'll talk through some of those methods. I'm not giving you all the maths of the methods because I myself am not a mathematician. You can ask Rob about some and other people. You can look to those papers if that's of interest to you, but it's more the application here. So here's just an example of a maximum parsimony tree. You're looking to resolve the relationship of these 5 individuals. You can see from this tree, it requires 1 substitution change, whereas this tree, you would have to have 2, so this is the most parsimonious tree. It's a pretty straightforward example. For maximum likelihood, you're estimating the tree topology that optimizes the likelihood of the data given a defined model. So I will go through, I should just say, if we run out of time in this bit of the session, we do have this parallel tutorial where we've sort of drawn upon, I've given you some examples from a wonderful Australian marsupial system of rock wallabies, some simplified data sets that you can run through some of these programs and sort of estimate using these different models yourself. So if we run out of time at the end, you can run through those. But there's a maximum likelihood example where you're estimating the most likely tree given the data, and in that scenario, you're not giving it a nucleotide substitution model, you're letting the program estimate the best model from the data. So there are programs out there, Rob's come up with one of them, to first estimate the most fitting nucleotide substitution model for your data, so that thinks about things like transitions versus transversion rates of nucleotide substitution. So it estimates that from the data, and then you estimate your tree topology given the data and the model. And so given this hypothesis, you estimate the probability of the data and the model of substitution with your parameters of theta and tau, so divergence, time, and population size. And then, I don't know how many people have thought about Bayesian. I always like these figures of like tree climbing or hill climbing. You're searching in space of these of this distribution or this of your so you're combining information of your priors for these parameters, and you're trying to estimate the likelihood, and you end up with a posterior probability. And basically, it's searching in space. And then when it finds a model that's better, that fits the data better, it moves to that spot in the search space. You find an optimum area, and that's your best tree. And it uses this Markov chain Monte Carlo method to search the space. And it gives you support within that posterior probability curve. I'm not going into detail here. You can ask questions about this later or look into those methods. So as Craig mentioned, there's different types of ways to analyse the data. So one approach is to concatenate your data. I'll talk through that in a second. And the other is thinking about this multi-species coalescent. And these sorts of discussions around what's the best approach for your data, they've stemmed through time in terms of looking at nucleotide sequence data, and it's the same for Snip data as well. So it's these constant discussions, and people are in favour of certain approaches versus the other. I'm not here to tell you what's right or wrong. I suggest people apply both approaches and look at discrepancies, because that's always kind of where the interesting stuff lies. Is your data robust to various methods to find the same answer? So concatenation is effectively stitching all your SNPs together to construct one sequence. And it's a useful approach because it can be fast and generally pretty accurate, except for, as Craig highlighted, under assumptions potentially of migration, recombination, things like that. But SNPs, you don't have to worry about the recombination. There's a variety of approaches that you can use concatenation data for. In the example in the tutorial, it's a maximum likelihood phylogenetic reconstruction. However, it can be problematic because it's assuming, I should say, you can separate different models of nucleotide substitution to your data. You can partition it. But if you don't, then it's assuming the same model across all your sites. But we know the genome's heterogeneous, so this may not be an accurate assumption, and particularly where you've got incomplete lineage sorting, where you've got different histories for those loci, so it can be problematic. So it assumes, yeah, as I said, that there's the same coalescent history of all your markers, and it can produce branch length errors, especially near the tips of the tree, if you don't take into account invariant sites. So I have to admit, the data I've worked with is largely sequence data, and I often pull SNPs from that data. I haven't gone from DART data or DDRAD data without mapping it to references, but I think Arthur probably knows about pulling the invariant information and how you deal with that when you want to concatenate from the DART tags. So you can touch on that later, maybe? Yeah. But the idea there is if you can pull in that information from surrounding those SNPs, that's always useful if you're going to be applying concatenation type approaches. So yeah, as I said, it's better if you can apply the concatenation to the original sequence alignments from which the SNPs are extracted. So not just the SNP, but trying to include some of that invariant site information. And there are ways to minimize or account for that bias, which can inflate the branch lengths. So there's an example of that in the IQ-Tree2 tutorial. I thought this was just a nice depiction of all the processes that make resolving evolutionary history quite complex. So we know that the genome's heterogeneous, we know that there's different mutation rates, recombination rates, there's different processes of selection, gene flow. It's not one thing is happening to the entire genome. Different parts of the genome are under these different pressures and histories, and this means that any SNP or gene tree doesn't always equal the species tree. This has been an ongoing problem, and we'll talk about tomorrow the benefits of mapping to reference genomes and trying to sort of understand that heterogeneity across the genome. But you know, these factors in terms of incomplete lineage sorting and hybridization, they can all influence our estimation of the species tree. So I'm going to talk now about so I've spoken about concatenation, but now we're moving to sort of the multi-species tree inference approaches. So this is using coalescent theory. And the coalescent is a mathematical concept, and it's supposed to represent the stochasticity of evolutionary history of a sample. So here, actually I'll go over it on the next slide. But what it's doing is it's looking backwards in time to allow ancestral inference. So this has been revolutionary. It's really bridged the gap between population genetics and the phylogenetics continuum. So here, and what it is you have these traditional models like the Fisher-Wright model of population genetics, and these are all the individuals, for example, in a population and how they're passing on their genes. But what we have is just a small subset of sampling that we're using to sort of represent and try and understand the divergence history. So you can see on the left here, those are highlighted within the broader population. On the right here, this is the reconstruction of just those individuals. And that's what we are getting. We're just resolving the divergence history for those individuals, and we're assuming they're representing kind of the broader history. And it focuses on the sample and it traces its genealogical history until it reaches a most recent common ancestor. That's what that stands for. And so what we're estimating is the time of divergence, or the time to when they had a most recent common ancestor, and so you get this estimate of time there, and we're also estimating the effective population size. So this is what coalescent theory does. So the multi-species coalescent, so this is done at an individual gene scale. Then these multi-species coalescent models, they extend this single coalescent to the case of multiple species, and it integrates species divergence with processes of drift and mutation, and you end up estimating the divergence time and theta, so the effective population size, and you get that for each population or lineage, as well as the ancestors, and you calculate that back through history. There are two ways of doing this. You can estimate the individual gene trees and then estimate the species tree simultaneously from that, or you can do it all together in one analysis. So the methods that I'll talk about later actually remove out the need to estimate the gene trees. So there are multiple ways to estimate the multi-species coalescent, so your species tree. And I guess one of the challenges with all this amazing data that we want is sometimes it's too much data to do the analyses. So sometimes when you're using, like for example, Snap, which is a full Bayesian approach where you're estimating the species tree, speciation times effective population size for these biallelic SNPs, it can be really computationally intensive. So sometimes people reduce their data set down, they look at subsets of the data, they might run replicates. So there's always this challenge of having the most complex models to estimate history, or using all the data, or trying to find some balance in between. So I've always found, although it's also Snapper now is the most recent program to run this full Bayesian approach to estimate the species tree, it is really computationally intensive, and I've shied away from it because I just haven't had the computer power. So not to put everyone against it, it's a great program. It's just that there are challenges and limitations with having these large SNP data sets. So we don't go into this in the tutorial, but I've just got it here to show you that there is a full Bayesian approach to estimate the species tree from your biallelic data. And as I said, you include in there your model selection and so on. And an extension of that is this BFD, Bayes factor delimitation. So as well as estimating your species tree, you can estimate the actual species. So these models, what they do is they lump and split taxa and reassign taxa, and they look at the support given that data for the different species and lineages, and you end up with a probability of these are your particular taxa and this is their phylogenetic relationships. This was quite challenging, and what they had to do, because comparing different models, the marginal likelihoods, they couldn't do that until some really smart people figured out how you can proportionally adjust them so that you can compare them. But this is one approach that if you're not quite sure of defining your lineages, and Upper will talk through also defining lineages in another way, but this is just one method where you could put in your data and it can tell you and define your lineages and assign them to a species tree. And this is just one example from the literature from Adam Lachey, which Craig mentioned with that review paper, where they've done that. So they modelled different combinations of different taxa and different individuals within those taxa to try and estimate what the true species were, and they found that the greatest support, so the best log likelihood value, was ranked number 1 here. So they've done it for 129 SNPs and 1,000 SNPs as well, roughly, and they found that it's this scenario here where there's a split of these two lineages from these 3 individuals. So this is the sort of thing that can be done using this method. I won't talk too much to this because Craig kind of touched on this a lot, but it's just always good to have at the back of your mind some of the limitations. Sometimes we just can't get around it, but trying to understand your system, trying to understand gene flow, trying to understand how representative your sampling is, the biases, missing data, just keeping that in mind when you're inferring. We're only as good as our data, like our results. Sometimes we have, known unknowns and unknown unknowns. So sometimes we're aware that there might be a lineage that we're missing, or we've got one individual that could be its own thing, or it could be part of a continuing cline that we haven't sampled well. So just having knowledge about that's really important when we're interpreting our data. So this is a method that I've been using more and more, and particularly with Snip data, I think because it can use all of the data and it's quite quick and efficient in terms of getting results. And I'll talk through this later, I won't go into detail, but basically this SVD quartets infers the species tree without needing to estimate the gene trees. So it is less computationally intensive, and it uses a full likelihood approach to infer the species tree from Snip data. And they basically analytically integrate gene trees out of the equation. So I'll go through this example a bit later, but this is really a valuable tool, and they have also shown that Snip data and full gene sequence data are pretty concordant in their results. I'll talk to this later, but there are advantages. You can look to their papers, but it's statistically robust under general models of molecular evolution and the coalescent. It produces species tree, and you get support with bootstrapping. And it seems to have the potential to provide more information for relationships deeper in the phylogeny because of this quartet-based approach. And if we get time at the end, I'll also go through this other method, which is called Miix, which one thing we don't touch on in this session is actually phylogenetic networks versus these bifurcating trees, which we know history is not always like split with two branches and no gene flow between them. So this is one method that tries to account for gene flow in trying to resolve the phylogenetic relationships. But I will add that although it's a great method, it's really looking at those relationships of more recently evolving lineages. So you probably wouldn't use this approach for deep phylogenetic reconstruction. But basically, Miix, it generates the maximum likelihood tree, and then you give it different models or different numbers of migration events, and it estimates them on the tree, and you can compare between those models. Yeah.
I was just wondering if, how far is deep? Roughly. Thousands of years, or
No, I mean, so I've used this for things that are a couple of million years diverged. I think you have to kind of take it with caution with other approaches as well. I don't have an answer of like a cutoff, sorry. Yeah, but just keep in mind that, this is one way, I've always used Miix with other methods as well. But one of the limitations is, trying to estimate this deeper in the phylogeny, you probably should be using a different method, or the simulations like what Craig was talking about earlier. So here's an example just to highlight from Miix, where they're estimating this maximum likelihood phylogeny. It has this drift parameter in there as well, so that's why it's kind of moving out. It accounts for drift. This is a human population phylogeny. And then basically you're getting this residual fit from the tree. So these are all distances between each pair of populations divided by the average standard error. And so residuals, the colours here, that are greater than 0 represent populations that are more closely related to each other in the data than in the best tree. And so they're kind of a candidate for admixture. So in this example here, based on varying levels of migration in these models, they found that actually there is evidence or better models accounting for migration, and in this case there's quite a few models of migration. I think one of the challenges here is always the more migration events you add, the more complex the model. So again, it's kind of thinking about, is your data, do you have enough data to sort of support all these models? But this is just to show you there's this method out there that's quite a useful tool. And then lastly, I just want to touch on thinking about divergence estimates of species trees. So I'm one of those people who really like to just see the topology. Although I'm interested in the branch lengths, I kind of want to estimate divergence times after. First, I really just want to see how those things related to each other, thinking about biogeography, biogeographic barriers, and so on. But then you want to think, okay, well, is that associated with climatic fluctuations in the Pleistocene, or is that a deeper split associated with the Pliocene? So historically, people, they either use fossil calibrations, if you have them, and put bounds on the nodes. This is kind of the beast type dating, or they estimate divergence times from the data. So with this Snip data, I'll talk through an example later using QAge is the analysis that's part of like the SVD quartets package. It's in PORP, but it's not SVD quartets specific. But basically, it's estimating the divergences using a maximum posterior estimator. This is really complex, sorry, I can't even say it. But basically, you're estimating the divergence times from the data. And so what these methods, these are for the multispecies coalescent methods, where you're estimating the speciation times in addition to estimating the topology. So in this case, this QH analysis and SVD quartets, it operates under this multi-species coalescent assumption. The nice thing about this method is you can actually input any phylogeny into this program. You just give it the species tree. So first you have to calculate your species tree. However you want to calculate it, you can then give it the species tree just as like a Miix format, like with your brackets and so on. And then what it does is it computes these composite likelihoods on the fit of the observed site pattern probability. So I'll explain this site pattern probability method later. This is just to highlight to you that using this approach, you can estimate your tau and theta using this map estimation. So the one thing to note with this divergence estimation is that it's got some kind of big assumptions which may sit well or not well with people, but it's assuming this JC69 substitution model that may not fit your data. It may not be a complex enough nucleotide substitution model. So it does sort of simplify the model for estimating this divergence time, and it also assumes a constant effective population size. And we all know that that may not be the true model for your phylogeny, but it's a good method. It's just, it does have some limitations.